Okay, welcome back. Just a reminder, you've probably looked at the functional groups and learned that different things are added to hydrocarbons to make them slightly different, whether they're polar or nonpolar. Today we're basically looking at building molecules and breaking them down, the biological molecules. So remember that carbon has 4 bond sites, and normally carbon and a hydrogen, so if I made methane, so let's look at this, CH4. Because of the electronegativity and the shape of that molecule, that is a nonpolar molecule. And if we took ethane, which is the 2-carbon molecule with hydrogens, and if we left it as hydrogens, it would simply be a nonpolar, but if we added that functional group of a hydroxyl group, an OH added to the end, that now creates ethanol, which is now a polar molecule, so that can actually be somewhat dissolved into water, because remember, water is a polar molecule. Before we get going on to more things, just a reminder, I use a lot of the Roddy speak, and biology being all those terminology things, remember that sometimes when you see something, it triggers other stuff to happen. So, example, if I saw, remember we talked about carboxylic acids? So anytime you see like a fatty acid or an amino acid, if you see the term acid in language, especially in the biology side of things, think of a carboxyl group. It probably has a carboxyl group attached to it. So lactic acid, which we'll talk about later on, or lactate, which is also lactic acid, that has the carboxylic acid group. Or if I see something like fatty, I'm going to think carbons and hydrogens. I'm going to think lots of carbons, lots of hydrogens, because that's a good stored sense of energy. Another little word, anything that ends in an ose, O-S-E, I would be thinking sugar or carbohydrate. Okay? Saccharide, also, saccharide means sugar, so those are another. And the other one that I would think about, always remember, if you see A's, A-S-E, think enzyme. Okay? As we talk about enzymes later in another lesson, whenever you see an enzyme, quite often it ends in A's or sometimes sin, and often the word tells you what the enzyme does. So it's always things like that to remember. As we build more and more into your biological language, the more you're going to be using these terms. Okay? So we'll continue, and then slide when we're building and breaking polymers. So when you build a polymer, we give that term either dehydration synthesis or also known as condensation. And if you think about it, when you put things together, you're going to make water and you're going to synthesize or put it together. So an example would be, I'm just going to put something like this. There's a hydroxyl group at this end, and I'm going to join it to something like this. If I join those two together, we're going to dehydrate synthesize, and if you look at it, dehydration means we're going to produce water or remove it. There's an H and an O, and if I take another H and I join those together, I will get water, or HOH. And then when I have it formed at the end, I have 3 carbons. What's left is, remember, there's the O. So I'm going to put the O there, and then 3 carbons. I didn't put any of the hydrogens in, but there you go. Now you have your new molecule formed. So quite often when you're like joining 2 glucose molecules, which we'll do in the next lesson, or joining a phosphate to ADP to make it ATP in another guide, making a triglyceride, you're going to go through dehydration synthesis. So every time you're doing dehydration synthesis, make sure you're looking at what you can join and how you can join it to make that water molecule, okay? The opposite of dehydration synthesis is hydrolysis. So if you look at the term hydrolysis, hydro meaning using water, it's just a simple word, like whenever we talk about like hydroelectricity, oh, that's electricity generated by water. So, and then lysis, when you think back to grade 11 or anything, when something is to lyse, it means to break. So the lytic cycle of the viruses, basically you're ripping open or you're breaking open the cells to release more enzymes. Or a lysosome inside of a cell, that means it's going to digest or break apart the cell. So hydrolysis just means you're going to take something and you're going to break it up using water. So I'm going to use a amino acid, which we'll talk about in another time, and I've got a dipeptide here, so I'm going to go, and then we'll just do an R group H with the amino at the end, and then this is joining with the amino, central carbon, R, H, and your carboxylic acid. So if we add water to this, and we have the proper enzymes to do it, basically that water is going to go and break open that peptide bond, and then the amino acid, I'm just going to write one of the amino acids, is going to go N to H, H, central carbon stays the same, H, R group, and then C double bond O, OH. So notice this N group here would gain one of the H's, and then the carboxyl group here is going to get the OH and the H. So now you've got the OH and the H, and then on the second amino acid you would get in the second amino acid you would get NH. The H coming from the water, central carbon, H, and then an R, and then you have your carboxylic acid intact from before. So that's where you get that extra H coming in, and no longer do you have a bond, a peptide bond, you have two separate amino acids. So you've got this amino acid and this amino acid from your original dipeptide. Okay, so hopefully that makes a little bit more sense. As we go on to the other lessons, the building and the breaking of polymers will become quite evident. Okay, we'll talk soon. Bye.